All right, we are live. Good evening, everyone. Emory Moynagh here for In His Image, joined by the inimitable, the great, the wonderful Sal Cordova. It's the first time Sal and I have been together on a stream for months, which is just like incredible. But it's good to have him here. I know his life's been crazy busy, and then I've been in graduate school, and then there was the holidays, and then I was sick, and so the whole. But we've got it together here tonight. We got him here, so please give him a warm welcome in the chat. And thank him for taking his time to teach us the things he knows about genome biology. I will try to remember in the description of this after the video goes up to put a link to his channel and his website and the Save McLean Bible website, which I know is in his domain as well. But without further ado, Sal, welcome to the show. Thank you for coming. Appreciate the time.
Thank you. This could be a very short show because my specialty is more in protein biology, and most people involved in protein biology focus on only like one protein if they're really good at what they're doing. And right. I'm not even good at one. I'm kind of lousy at 4 or 5. But that's just so. It may be amazing. Everyone gets so specialized nowadays because there's so much to learn in biology, and it takes so much work to be a researcher. To actually move forward. So we have guys that their specialty is just like knowing an experimental method. And they're going to spend a whole year trying to set up I remember talking to one of my professors. I said, boy, I'm so jealous. And I had classmates that were experimentalists. I said, I'm so jealous of you guys. They said, don't be. And my professor, he was saying, yeah, sometimes it takes 2 months to get like they're purifying a chemical. He said it took 2 months, and you hope you get it right.
Yeah, and this is why I have like
chromatin.
Yeah.
All the amplification and purification steps. And he said it could be really frustrating when it doesn't work. You're holding that one test tube sample.
Yeah.
And
you pull up this one test tube with 30 milliliters or whatever of liquid in it, which is the entire month's worth of work, and it's not pure enough. We got to do it again.
Right. So, and then they might be testing one residue because he was trying to do chromatin studies in chromatin states. So you have a post-translational modification on a single amino acid, and it's like finding a needle in a haystack. And so all this to say is that I'm very flattered that Emory asked me to do this show, and I'm just like, I'm so unqualified, but hey, I get to hang out with him. So even if I do a lousy show, I get to see Emory. So guys, just to let you know, to me, protein biology is exciting because you get all the cool machinery and you get all these cool animations of what goes on in the cell, like the ATP synthase,
right,
and ion channels and stuff. And this is really painful for me to admit, that I hate genomics and genetics. And I worked for a geneticist for 7 years, and so I had to just suck it up because he gave me a wonderful opportunity.
Right.
And it was great when I had probably my lifetime achievement in secular peer review happens to be a genetics paper in population genetics, which I loathe even more. And it's not that I was very grateful for Dr. Sanford coming into my life, but it's like you just look at ACTG, and it just goes. And I remember the day he hired me, he said, "Look at this cell," and he just showed me these screens of ACTG, and just like, oh my goodness,
what did I sign up for?
You're really serious into this. No wonder you're. I think I'm gonna die.
Well,
look at that's interesting. This little pattern here is 3 letters. I'm just like, okay, so go look for that 3-letter pattern in the genome and see how often it repeats, and look at all the properties. And we'd add little colors sometimes.
Yeah.
And I'm like, I'm gonna do this for a whole year.
Yeah.
So, so at least with protein biology, you get to see kind of, maybe the way to liken this is if you're building a building, you have to study the blueprints. In the olden days, we had blueprints. You can either appreciate architecture by looking at that beautiful building, walking through it, and just marveling at the architecture, or you could study the blueprints. And that's really kind of what it was for me. On top of that, then most of the interesting characteristics that are cutting edge now in gene research is the non-coding regions. So most of my genetics has been in the coding regions. So I may be a total non-help.
Well, today. You're probably a total help because I have had no training in how to use any of the algorithms or anything like that. I've downloaded some of the phylogenetic and genetic algorithms. Like I have Eugene, I have Mega, and I know a little bit about how to use them and understand. I understand how to interpret a phylogenetic tree because that part I figured out on my own. But like, I can look at those A, C's, T's, and G's and go, okay, I can see some things here. Like I can see, I'm starting to be able to visualize the alignment a little bit on my own, but I don't know how to use any of the tools. And I figure most people out there, creationists and evolutionists, probably don't know how to use the tools and definitely don't know how to interpret the results they get once they get the results. So that's kind of what I'm hoping for here. I even went out and bought a book on it over Christmas as one of my Christmas gifts to myself, on how to, on the subtitle is Concepts and Applications of DNA Technology, simply because I'm trying to figure this out and improve my understanding of this. And so I figure I'll let someone who knows a lot more about it than I do teach me about it. So
I'll try not to disappoint. This one reason I wanted to have this conversation, and we can try to explore this a little bit, is I think you overheard a show I had with Rebecca Davis, examining evolution and Bread of Life, or even my channel, Evidence and Reasons. And I might have been talking about evidences for universal common ancestry. And so I was saying that, and this is good for our audience to maybe to hear this, because I don't think it's appreciated enough, because it nearly broke me. And thank God I was working for Dr. Sanford at the time, and I was surrounded by creationists, because when I went to bioinformatics, I said, if I did not have that background, I think I would have lost my faith in creationism, or I would have become like a Michael Behe theistic evolutionist, which probably wouldn't have been quite that bad. I mean, Michael Behe is a certain kind of theistic evolutionist compared to someone like Swami Das.
Yeah, Behe and Swami Das can sit on the same stage under the same label, but they're not the same thing.
Right. So I just wanted to clarify that. So, I mean, I think calling Michael Behe creationist is at least in the correct spirit of the thing, in that he believes that there's kind of this mystery of where complexity emerged. So belief in common descent does not necessarily mean you accept that it can be just mindless.
Yeah, so I would say that calling Behe a creationist is more accurate than calling Swami Das theistic, in terms of I didn't necessarily mean that in terms of their view of God. I meant that more in terms of their view of how life got here. But that's another.
Yeah.
That's another.
So
that's a whole other kettle of fish.
where was I going with this? And this is why it's important to know gene browsing. And this is how it got me. It really made me think. We'll get to the technical stuff for eventually, but in the Maury show, they always had these things where they doing paternity tests. And, at the end of the show, they have a couple, and the guy's always like under the gun whether he's the father or not. And at the end of the show, Maury will reveal, you are or you're not the father, and then one of them does backflips because they're so happy about the outcome.
Kind of like watching a train wreck. It's terrible, but you can't take your eyes off.
I know, it's just terrible how we get so entertained by other people's misery, and unfortunately, I enjoyed it, not because I would never necessarily turn it on, but you know how how it is. Sometimes the TV's just on, and my mom would just have these shows because there's just nothing else to watch. And the way they're able to determine paternity is with short tandem repeats, which are, I think, real mistakes in the genome. That they're novel enough that they'll identify lineages, and if the child has shared mistakes with a parent, then with a dad, then that's good evidence of paternity. So by way of extension, not using short tandem repeats, but rather things like pseudogenes, I began to see shared errors between primate species, and that did shake me up. So I'm now comfortable. I just wanted to get this off my chest. For a Christian who believes in the fall, the literal fall of Adam, and that God said the ground will bear thorns and thistles, and that death entered the human race at that point, I would presume that then death started to enter the animal species, most of them. The Hydra seems to be immortal, and that's kind of an interesting.
Yeah, there's a whole show we could do down in there.
Huh?
So.
What?
There's a whole show we could do into going into the Hydra genome and trying to figure out what on earth is going on there.
Yeah. So it's like, okay, so all the species under the planet then got reformatted simultaneously. So there's it's kind of a second kind of wrathful, miraculous event that affected biology. So we had the first special creation, and then death invading all species, or at least all animals that I know of. And so I'm comfortable with the idea that there are shared errors, possibly because of that. There could be shared errors because there are genomic features that just optimize certain regions to be kind of fragile. And that would be my explanation. If, as you know, even from an empirical standpoint, if we do show that the fossil record and life are young, then it's a done deal. There's no common ancestry. We have to, as a matter of principle, say that the shared errors need another explanation than common descent. But that's definitely a tough road, and I'm willing to take that road because of the miracle of complexity. And after seeing James Tour just rip down Dave Farina, it's like, yeah, there is a God. There's just like no question about it now. And people like Change Tan pointing out the difficulty of eukaryotic, prokaryotic evolution. It's like, okay, I think the problems on the other side of the aisle for the evolutionists and the abiogenesis proponents, I'd take our battle over pseudogenes any day.
Yeah.
And then I also I had to do a little bit with the ALU elements and repeats in general in the human genome, because there's huge amounts that it's turning out most likely to be functional. We also know that a lot of pseudogenes probably have some regulatory role. And they probably aren't genomic mistakes. So there's a lot we don't know, but I do think there are some shared errors across species. One thing that would be valuable, and Lord, if we only had enough days in our lifetime, I think you and I could find some interesting patterns, because if we share errors with gorillas, quote unquote, that we don't share with chimpanzees, and then also if we share errors with chimpanzees that we don't share with gorillas, I'm pretty sure that that's a very problematic thing for.
Yeah, that would raise significant questions about the official narrative.
And I actually think I saw that in the study of the gene that I mentioned on my show with Rebecca when I talk about the Interferon Lambda 3/4 gene. And I was just looking through it, and I was like, some primates have that mistake and some primates don't. How do you explain that?
Right.
And so this would be how that relates then to what our talk. This is the one kind of cute gene browsing I was actually able to do that was of any, what I think, personal utility, because you could actually see the introns and exon boundaries highlighted. And now that whole thing's an interesting pattern. And I wanted to say, well, okay, if we were on the Maury show, how would he say that? Is, you know, that primate's your father, that primate's your father, or like, we can't figure out how this is possible.
Yeah.
How can you have two fathers, you know? So
yeah, that doesn't work very well, but that's neither here nor there.
okay, so let me share my screen.
All right,
and I'll show you what little I have on this. This is the Interferon Lambda 3/4. And how did I discover it?
All right. So what program are you, before I go any further, Sal, what program are you using to view this? How are you viewing this?
I don't know how I got here. And when I was doing my class in this, it was kind of the same way. I was just clicking around, and the professor said, look at Interferon Lambda 3/4. And just like, I have no idea where to begin. Or I think he threw me a sequence. He said, you go find it now. I think he sent an email, and he just gave me ACs, T's, and G's sequences, and this is what it looks like. He said, okay, have at it, guys. All right, so if this the reason I'm showing this now is if this blows up, at least you got to see what I did.
Exactly, right.
So let's see if I could rebuild how I got here. Okay.
Well, I can tell you part of where you are. You're on the NCBI NIH database. I can tell you that's where you are. And I think, yep, you're if you were to, and there's the accession number, the reference number right there, the NG number. So anybody who wants to go find this particular sequence can go find that now.
Oh, okay.
Because if you put that reference sequence into the nucleotide database on NCBI, you will be able to find that sequence.
And see, now I can't get rid of this stupid pop-up now.
That's the little X there on the bottom right.
Oh, see? All right, we both learned something. All right. So I got here, I think, and then it says highlight sequence features. Yeah. And what that helped to do was I could then go down here and it would highlight the exons. Oh, see, I lost the exons. All right, so when I clicked on whatever, it blew it apart. So let me try this again.
All right.
And we might have to just kind of crawl through this. So I press highlight sequence features. See there, and it puts that stupid pop-up again.
Okay.
But then it gives me, you can see it right here. The first exon is just ATG, which is methionine, or methionine, or methionine, which is the start codon. And then you have all the amino acids there, all the codons. And then when it is just kind of this uncolored region, that's the intron, and then the next exon. So this is the next exon. And then the next exon.
So what's being highlighted right now is one gene out of the sequence of nucleotides there. If you were to search for a different gene product, you would get different exons and introns, correct?
This is, I would believe that's correct. This is in a sea of, see, the first problem is this is a pseudogene. And like, what do you do with that, you know?
Yeah.
That's the first problem. And the other thing, this is on chromosome 19. So it's not like, the problem is, what do you do with that? Because it's the whole genome. It's not like there are any actual natural markers there to say, oh, look, here's a new gene. You know, it says to start here. You have to sort of infer where the start is. So
right.
where the actual gene begins in a eukaryotic cell can be, it's not quite as clear, I think. I mean, you could tell where the protein coding sequence starts, but we know the gene is more than that.
Right.
It has
Because
the
the gene
promoter
includes the
regions and
introns.
whatever, and that could be, you know, I don't think it's extremely well defined where the actual, you could say this is where the gene begins. You know that it's not quite as, like in prokaryotes, you have the Shine-Delgarno sequence sometimes, but in the eukaryotes, the way the gene is implemented could be very complex. That's the extent of my poor, pathetic knowledge on this. And there is, let me look at something. I'm going to look at intron. You can take this down for the time being.
Okay.
Back to this.
I've left your screen shared, Sal. I just haven't put it up on the screen right now. So if you want to change something, that's fine. So what I noticed there is something that I don't think really gets conveyed in genetics textbooks, and that is the exons make up a much smaller portion of that particular gene than the introns do. There's a lot more introns there than there are exons. The exons are much smaller.
It really is shocking to actually see it. Compared to what the bacteria and the way they present it, it's like 98% of the human body is the non-coding. The human genome is non-coding. I mean, you really see it when you're browsing like this.
Yep. Now, Sal, suppose I wanted to find a different gene. How would I, that is on that chromosome, how would I do that?
That's what I don't know.
That's what you don't know. Okay.
The other thing I need to point out is I'd imagine that a chromosome has like, what, 50 megabytes? Or
It's
50
not small.
It's not small. And so a lot of times when I was going through this, it would just lock up. GenBank would like lock up.
Yeah.
So what they do recommend is you download the sequence to your local computer, because they have kind of a metering algorithms or something there, because even though they make this available to the public, they don't want someone to just be able to just have at it all day long and use their computer time at the expense of other users.
Right. So, they're a database. They don't want you doing all the work on their database. They're just a database. They're providing the information for you to go, hey, go use this on your laptop on your time.
Right. So that is that's one of the challenges. Is when I took my bioinformatics class, we never got to the point. I mean, there's just so much in bioinformatics, and everyone has their own pet genome that they're studying. So if you're a microbiologist studying E. coli, or heaven help you now if you're a microbiologist or virologist studying COVID, you're gonna have tons of databases from all these regions, and it's gonna be almost identical, but you're just combing through it to find out that one variant. And you know, I feel for the guys that find like Delta or Omicron, and they're like, they probably have every gene sequence that was ever taken of COVID in every person that they sampled, and you're going to try to put that in a database, and now you're going to look to see for a variant. Yep. All right.
Try aligning that. See how long it takes.
Okay, so the bioinformatics class kind of had to address, okay, we know each of you has different genomic needs, and you will need different techniques. So for a lot of the guys at the NIH who are studying human biology, it's like, oh, we just need that one reference sequence. Right. And so the gene browsing tools you have are different for that. I just needed to point that out.
Okay.
So what we mean by sequence browsing could be very different depending on who you're talking to.
Right.
That's why bacterial gene sequence browsing can be good and bad. If you don't have to collect all the bazillion variants across the entire planet on one particular organism or virus, then you're good. You could just get like E. coli, like let's say you're trying to do a phylogenetic tree across select species.
Right.
You get the reference sequence, like say a big consensus sequence for highly conserved E. coli, and BLAST, mercifully, the BLAST algorithm is organized like that. It's not going to give you, if you take a sequence and you want to blast across species, all the defaults are nicely set. It's going to give you the reference sequence, like say for E. coli.
Mm-hmm.
It's not going to give you all the strains of E. coli. It's going to give you, so if you want to compare a human and E. coli, it just does that naturally, which is nice.
So you're talking about blasting a sequence. I have a vague idea of what that is, but what for everybody watching, because this is supposed to be kind of a tutorial, can you show us what that looks like?
Okay, so this is a bioinformatics professor said, don't ever do a live demonstration because it's not going to work. But given where we are, and we're just going to, I tell you what, we are going to give the audience a taste of what it's like to be a graduate student.
So you get to live in our worlds, all of you get to be sucked into the lifeless world of a graduate student.
Okay, so you could bring up one of my screens here.
All right, let me pull your screen up real quick. There we go.
And let me see. I'm gonna yank a let me just try to yank this sequence here, and I'm gonna clean it up.
Right.
So I'm just cutting and pasting into a notepad. And sometimes it has the numbers there, and you can just clean it up. And the genome doesn't have spaces in it. That's just kind of there to help.
That's there for us.
Yeah, that's there for us. I'm just going to copy it. Now I'm going to go to, I just type in the word BLAST in the internet. And the nice thing is, it's amazing that when you type BLAST, it doesn't go explosion or anything. It takes you actually to a genomic tool. I mean, you think of all the things that Google could look for, but usually it goes to the NIH here, and to this basic local alignment search tool. So if you happen to have a DNA sequence, you could just kind of put it in there and say, just blast it. All right, so I just use all the default parameters, and it's just going to search the database. It's just going to go there and search the database for like all the species that may have that. And I have no idea how this is going to
I suspect this is going to be a long blast, because that is a sequence that is a primate common, if I remember.
Who knows where it's going to search, but what it will usually do is, like it'll say quit after like 500 identifier, identifications.
Right.
So you could take this screen down.
Okay.
And
of course, the minute I do that, Sal, it's going to find it. You know that's what's going to happen.
So,
go ahead.
But there are some things where this is actually how I stumbled on a paper, and I was doing the work, as many people know, on nylon-eating bacteria. And I was saying, John, I don't know how to do any gene browsing. He said, let's go ask so-and-so. And I was just really shocked. A lot of my professor colleagues actually did not know. And it's like I said, why was this? Because they're specialized in what they're doing.
Right. And unfortunately, as creationists, we can't afford to be specialized. There's too many holes that need filled.
Right. And I had worked with Jeff Tompkins on a human genomic project, and that was actually really difficult because that was like when I first started my job with Dr. Sanford. He assigned me to help Jeff, and I really didn't know very much at all.
Mm-hmm.
And the frustrating thing is, if you work at the NIH, you could sometimes go down to your colleague down the hall and say, hey, I don't know how to do this. Do you know anyone who could do this for me? And they'll say, yeah. And he'll just sit you down there for 2 hours and you'll learn it.
Mm-hmm.
Whereas, the way we have to do it is I had to go to class, and then maybe I could build a relationship with my professor, right, and I could have those questions answered. So, now this is what I wanted to tell you. Emory, you're a graduate student. Do you have any colleagues or professors you can kind of just like knock on their door and say, sir, I'm Emory Moynagh, and don't hold it against me that I'm a creationist. So you wouldn't say that. But how do you do gene browsing? So, I mean, what would you do gene browsing for? Would it be human or animal biology? Or
well, my research work is not anywhere near human. I stay away from humans. Like the whole point of me not going into medicine, because my parents actually suggest, you'll be a doctor. No. But like I stay away from humans. I even stay away from the whole human chimp thing, to be perfectly honest, just because I don't have any interests. Like my interests are in plants, in insects, in zoology, maybe. Polyploidy is my primary interest. I actually have framed up here my polyploidy paper that I have published. It's under my real name. That's why I didn't show you the whole thing, because it has my real name on the front. But my interests are just not in human. But like my kind of goal with this, I want to be able to do sequence comparisons, and also this could be useful for baraminology, doing sequence comparisons and that sort of thing. So that's why that's part of the reason I want to learn it.
Okay, so I mean, this is helpful to know. And you could use the BLAST. BLAST is kind of a nice start. Now, I talked to some evolutionary biologists, and they kind of poo-pooed me for that, which they always do. No matter what I say, they're going to. So I take that with a grain of salt. But I have the results here. We can look.
All right, 1 second. Let me pull those up. Oh, my.
So we have these 500 hits. And see, like, we had this... we have a lot of Homo sapien hits.
It looks like a lot of Homo sapien on there. I don't see. Is there anything that's not Homo sapien?
I saw Hongo somewhere down there. Gorilla right here. And see, we're 98% similar to the gorilla.
Mm-hmm.
And I started looking at this. So let me look at the table here. So we have the accession number.
Right, which for those of you who don't know, that's how you find it again if you need it. You write the accession number down.
So I'm going to look at this Pongo. And I don't know what, I think that is a...
I want to say that's the Gibbons, maybe? Where's Erica?
Where's Erica? See? So I'm just going to see open link and new tab, and let's see, that way I don't lose the original. So interferon. So this is what's going to be aggravating is in human biology, it's IFNL4. And in the gorilla, it's called IF1IA2.
Okay, so this is orangutan. I just looked it up. It's orangutan.
Oh, wow.
Pongo is orangutan. This is the Sumatran orangutan.
Okay, so now let's look. And there it is. This is.
So this is the whole sequence. This is not just.
That's hard to believe. That's tiny.
Well, no, this is the whole sequence for that gene. I think this may not include the introns. I don't know.
Yeah, this doesn't look right. I got it. I think. You know what? I think this is just the exons concatenated. Honestly.
Yeah, I don't think that's the introns, Sal. I think the introns have been left out of that, and that's just the exons.
And we can let's see if it says highlight sequence features. Does it allow this? It's going to highlight the whole thing. I bet. Yeah, there it
Yep,
is.
whole thing. Yep. Yeah, it's cut the introns out.
Okay. I just learned something, so that when I look at like the topoisomerase, which is this one that I study, and since it's, we're so obsessed over the protein, we rarely actually study the gene that codes for topoisomerase.
Right,
That one has 35 exons, 35 exons.
35 exons.
And I think we go for the main alternative splice, so to speak. I don't think I don't think a lot of people want to touch all the alternative splices of something with 35 exons.
Do you realize how many ways you could splice that?
Yeah.
Yikes.
Yeah, we're fearfully and wonderfully made. So how do we ever see, this was interesting that I think when I ran see, this one just gave us, I gave kind of like the exon, one exon, and so it's actually now going and linking to full entire, the whole gene database,
Right,
not
the
the
whole
database inside the genome. And I actually don't know how I managed to stumble upon the chromosome 19 sequence, because sometimes BLAST will take you to a database that is just really more a protein database, and other times it would take me to the database where you're browsing the entire chromosome and you're lucky that it puts you in the right spot. I don't know how it's been now how many years? 4 years ago since I was tinkering with this. So
see, okay, so Sal, one of the reasons that I wanted to talk about this is a paper just recently, last year, a couple months ago, I think, came out from Gordon Wilson, and it basically proposed using taxonomically restricted genes as potential identifiers for barometers. Now, I don't think that's quite going to be quite perfect, but I wanted to be able to at least find genes and compare similar gene sequences to look at similarities and differences and so on, and see what I could learn from it. Because up until this point, I've basically just been using mitochondrial sequences, which are not as good.
Well, it's funny because if you just happen to let me point one thing out. Can you hold that thought for just one moment?
Sure,
and
sure.
If we can drop the screen. Do you see this one here? I threw in this. I threw in a sequence, and you get a lot of 100% hits. It happened to come from this one here, because I happen to have the accession number on chromosome 19. And let me open it now. This is going to be interesting because I took this sequence from somewhere, ran it in BLAST, and let's see if it takes me back to where I'm hoping it will, or maybe even a better place. So Homo sapiens interferon lambda 3. Now this is also, I mean, maybe I'm not telling you anything you don't know, but the same gene could have so many different names, and it may not even correspond to the protein it codes for, and that can be a nightmare. So I'm presuming you'll be studying genes, not non-coding sequences. Is that correct?
The way Dr. Wilson had wrote about it, we're talking about things that are actually protein coding, not non-protein coding.
Okay, now, so this is going to be interesting because, all right, this is interesting. Let me point out something. You see how short the sequence is?
Yeah.
For the pongo, for this orangutan, but then you compare it here, you can see how big this is. And it said that there is a 198% match. And you're like, how could that be? It's because it took only a small piece of this.
Right.
And it's going to say, okay, there's 98% match, which is interesting because this shows that this whole 98% similarity, it's like, well, it's like taking two books and comparing all the words in them and saying, hey, they're 98%. Look, THE, the word the, is 100% identical in this book with that one. And
all it means is that that sequence is contained somewhere in this lengthy thing. That's all it means.
right. So the way it's laid out could be different, and that actually is problems for common descent, because when you actually see how it could be laid out among chromosomes, and we can also, we need to maybe just have, before we complete the show, we need to have a little discussion about sequencing technology, because that can affect the results. But okay, I'm going to do a highlight sequence features. Here, now let's see if we get better exons and introns. Yeah, this is a little better, because the last one didn't give one as good. And you see here, now it gives you the TGA, that's the stop codon.
Right, yep.
It's really kind of scary, Emory. You and I are obviously grad students when we know the spelling of start and stop codon. We're starting to read this like a foreign language. It's kind of scary, brother.
Oh my. Yeah.
I think I need to show this, replay this on our channel, just so people could see what nerds we are. But just to get some street credibility. So there's a start codon there. And then now let's compare it to this one here, where we started all this. Oh my, see, I may have lost the original. Let's see if
Sal, I'm pretty sure you were on the original.
No, that wasn't it. This is the original. And you could see here in the original where we started off. See how it cuts off? It doesn't give me it doesn't even give us the stop codon.
There's no stop. Yeah, there's no stop codon.
So this may be a point. You can take all this down now, Emory, for the time being. This is educational in and of itself. It's like, don't trust where what you may stumble on may not be the whole picture. And you don't know what you're stumbling on, because when I ran BLAST, it took me to where I really wanted to go, where I get a better view of the entire, where I go from start codon to stop codon. Whereas when I first, when I went in there and typed IFNL4, that's where it started me on, it took me to there where I got an incomplete sequence. And like,
because it said INFL4 in the title, and BLAST, when you search by the name of the gene, it searches the titles. Whereas if you BLAST a sequence, it searches the sequence.
Which was interesting because exactly. So some of the records are like that. So they may have formed this record IFNL4, and it's incomplete or there's problematic. I think let me read. Let me see something here. I have a little. I had some notes on this. IFNL4 is the exon sequence supposedly, but the gene is IFNL3, but they're really supposed to be the same thing. So how could that happen? I'm giving you a tough lesson in how the databases are and how the reference data is assembled. You could have, it's always constantly under review because sometimes you have to strike entries from the record. And you got to be really, really careful to, you don't know, some of this is done automatically with all the homology and namings, and you don't know what you're going to get.
Right. And so actually, I want to point out something. The title of the Pongo sequence you clicked on was the predicted sequence, which means I don't know if that's an actual sequence or if somebody has gone, okay, they're related to humans by this long ago, so they should have this sequence of DNA. Now I'd have to look at it again and look at it, so I don't know.
That's kind of hard because sometimes they say it's predicted based. I don't know whether it's a protein prediction, meaning they add the DNA and they're trying to predict.
Meaning they have the protein and they've worked backwards from the protein.
Right. when they say it's a gene prediction is saying, okay, we have this sequence that we assembled.
Okay.
And if they assembled it on a chromosome, God help us, because you know how they do that. I think like the chimpanzee chromosomes,
Yes.
they took a human model and then said, oh, since we're related, this one goes on this chromosome and this one goes on that chromosome.
Right.
And here's the synteny. And then they publish papers. They say, look how similar our synteny and all this is laid out. And just like, oh boy, garbage.
Yeah. It's a garbage number.
Absolute garbage. Okay, so now this is a good time to talk about, for the benefit of our audience, the problem with gene sequencing is sometimes they can pull out an individual chromosome, which is nice, and then you throw it into your sequencing process, and it chops it up into a bazillion pieces that are only like 300 bases long.
Yeah.
Depending on some of the fast sequencers, only 100 bases long. You're hoping and praying that when you reassemble this puzzle, that you assemble it in the right order. And so you have to have some evolutionary assumptions sometimes to guide you to how to put this together. Now, why is that? There are chemical issues that make it very difficult to actually pull it out in terms of like 30.
Right.
I think the longest sequence strand we can pull is like 30,000. And you got to pay big bucks to run this specific biotechnology, packed biosequencer that can actually go through that. The other problem is when you have lots of repeats, that messes up the chemistry and you don't know where something starts or ends. So when you look into, especially for the human genome, when they say there's this repeat there, it's of unknown length.
Yep.
So I don't know how in the world they're able to assemble something. You know, they say we've assembled the human genome. It's like, what do you do with all the repeats? You don't know how exactly, you have all these spaces. You're kind of guessing.
Right. You've put together something that vaguely resembles what the human genome actually looks like, and how vaguely is a matter of debate, and
Right.
we may never quite find out.
Right. So this is why it's kind of nice if you're studying specific genes where you have the protein, you have at least a reliable guide.
Right.
You can estimate where the exons are because you have I'll tell you why, because we have mRNA data. What we do is when the cell's doing its thing and creating these these nice what do you call you have a pre-mRNA. What do you call the processed RNA that has all the introns pulled out? We're graduate students. We're supposed to know this.
Jeez Louise. I know exactly what you're talking about. Like it's not mRNA because that's what is the name of that RNA strand? I should know this.
I mean, we just call it mRNA, but there's a certain thing where it's matured.
Right. I know what you're talking about because this is post where it's been spliced and all the introns have been spliced out and they've spliced out any exons they don't want to.
CDS, a coding sequence? I don't know. I have it here on Wikipedia. They call it mature mRNA.
Mature mRNA. Fine.
All right. For lack of a better term. So they have there are these databases of mature mRNAs.
Mm-hmm.
And then they can say, okay, we'll assume that codes for a protein. Now that's kind of dicey because we're finding out that there are a lot of RNA genes. They don't code for proteins in humans.
Yeah, because not all RNA codes for protein. It does a bunch of other stuff.
Yeah, so that's another mystery. What are these mRNA genes doing? But so we have that. So that's how we're able to identify the exons. So there are regions that we can consider like, yeah, we have relatively high confidence. We've got this.
Right.
We've got the exons. Therefore, we define the introns, and we got this. But in between the genes, the intergenic regions, it's like,
Right.
Despite all the press, because they're trying to get congressional funding and all,
Mm-hmm.
and all the proclamations of victory that we got this, it's like I'm skeptical.
Yeah, the non-coding the coding sequences are probably fairly well established. The non-coding sequences, particularly the ones that are purely structural or just long strands of repeats.
And if we had enough days in our lifetime, brother, I found this one paragraph. I was searching on chimp and human similarity, and I know you're not that interested in it, but for the sake of the audience, especially those sophisticated, the centromeric regions in the chromosomes, they're not mapped the same way between chimps and humans. I'm like, one paragraph. I said, that's the darn most interesting thing. Why'd you invest only one paragraph in this?
The centromeres aren't mapped
And I wrote John Sanford.
And you put that in one paragraph? That's like a whole paper.
I'm just like, oh, I think the researchers didn't want to touch. They just realized they opened a can of worms. It's like, oh, we don't want to go there anymore. I'm like,
Yeah,
no. Okay, so you heard it here, and I passed it around to John Sanford, and it basically went nowhere because we're all just stretched thin, like unbelievably thin, like what that's. But I just like each time I stumble on, it's like, if we only had more creationists that really knew genomics, we could just light up the evolutionary world. I mean,
yeah, easily. There's so much stuff in the DNA that we just haven't, because we just don't have the manpower. They don't have the manpower. They don't have the manpower to go through the amount of data and they've got 95 to 97% of all scientists in the world, and they don't have the manpower to go through all that DNA.
Yeah. So I'm glad. See, this is why, brother, we can hang out, because you appreciated just now what I was saying. Centromeric, they had to have some sort of special sequencing techniques. I don't know how they did it. I don't know how they're able to do it, but they said we don't quite understand how the centromeres are. So I'm just throwing this out there because there might be some creationist that needs to hear this. And so when they're talking about, I mean, if I had my, if I had time and I could make a case, I'd start, when they talk about common ancestry, he said, look at all the evidence. I said, well, look at all the counter evidence. How about the centromeres being kind of switched? And I only got to spend like a week on the centromeres. I said, yeah, Dr. Sanford would want me to just move on. He's like, do the ERVs. Yes. I'm just like, oh, John. Okay, go from the allele repeats now to the ERVs. Yeah. And I found some cool things. There's just no end to this. So.
Again, right? I could spend the rest of my life in BLAST. I could write a paper probably every week on something I found in there, and I wouldn't run out of things to write about.
Absolutely. So, I've tried to give you all the caveat. It's kind of good to run through that. You could see how even BLAST will kind of lead you in strange places.
Right.
And I stumbled out and I should tell you this. I don't know if he's going to have time for me. I'm going down to the ICR January 21st. I'm not going to mention the gentleman's name publicly that I'm going to see, because it'd be better for his career. I'll tell you backstage who it is. Okay. But Jeff Tompkins works there.
Yes.
And he's Mr.
I've talked to Dr. Tompkins. He's a great guy.
And there's some other, I had lunch with Dr. Rob Carter October 2nd. The reason I know that is October 3rd is when I got trespassed from McLean.
I remember this.
And we had a show. Yeah, that night.
I also remember being incredibly jealous that you were getting to have lunch with Dr. Rob Carter, and thought about not speaking to you for about a week.
So,
go ahead.
Yeah, he took me out. God bless you, Dr. Carter, if you're listening. and he's so busy. I'd like to go hang out with him. He could probably teach me how to do gene browsing.
Yeah, he probably could.
So there would be Jeff Tompkins and Rob Carter. But then again, they specialize very much in human biology.
Right. They're both
So-
very. Although Carter's PhD is in like marine biology, so he would probably know how to do some stuff outside of human biology. I don't know what Tompkins is in. I off the top of my head.
Okay, so like Nathaniel Jeanson and Rob Carter, human biology. So they know all the human databases, like
Mm-hmm.
They know the Africans,
Right,
the Irish. I just can't imagine. I think I'd be sick of looking at every human genome. Again, it's just like looking at the bacterial genomes, all the ones, and I guess these guys would even look at like individuals and say, that's interesting. Look at that feature.
Well,
there.
I've seen Jeanson's YouTube series he did last year during COVID on the secret history of the human race or whatever it was. And like just listening to him talk, it's like my head hurts because like you're talking about like individual DNA sequences from individuals all over the world, mapping them onto this giant phylogenetic tree. And I'm like, whoa, slow down.
See, I got to tell you, I respect guys like that.
Mm-hmm.
I respect because like Rob Carter, John Sanford, Nathaniel Jeanson, Jeff Tompkins, that's what they do.
Mm-hmm.
And that's why I like protein biology. You don't have to, I would just look at that one topoisomerase. It's so beautiful. And, I don't have to compare it across species.
Mm-hmm.
And
see, that's actually what I want to do is I want to compare sequences across species for the simple reason that I think it would be very illuminating because it would, for one thing, it would show that there's a lot less similarity than maybe we think there is. For another, I think it could be very illuminating for baraminology, which is my pet project. So,
okay. So now, since we're talking about that, and I know a little bit more where you're headed, I think protein sequences are also very interesting.
Yes, I actually discussed this. I didn't get into this in the video I did with talking about Wilson's paper. I don't think. I'm trying to remember now. I recorded it before I got sick, so I don't remember now. But I actually think protein sequences might be more valuable than DNA sequences because there's more information. There's 20 amino acids to 4 DNA nucleotides. So there's going to be there's more possibility for variability at each, I don't know what.
I mean, it's just more evident.
Right.
The other thing is the reason this is, I'm actually glad we're having this conversation because you can either go the protein route or the DNA route. The protein route sometimes can find similarities you can't see in DNA. Just let me give you a little pointer here, because when I was searching for myelinase, I could not find gene homologs. When I did what I did here with the BLAST, with the nucleotide sequence, I got 20 homologs. When I did it with the protein sequence, I got thousands. Now that may sound shocking. Why is that? The architecture of that protein was beyond what they call the twilight zone of homology, meaning you only had, the twilight zone is anything less than 12%, but this one could identify it with 5% similarity in an alignment, which was shocking, 5%. But it found it. I mean, the features, you could actually just look at it because I'd actually go through. It's like, no, this is totally non-random, and you could actually physically see the alignment when you ran it through Mega. I'm just like, only 5%, but it's just like undeniable that there's something there.
Right.
And then also there's experimental evidence. So even when you had like 50% similarity, there was chemical evidence that would catalyze the same reaction. I mean, if someone wanted to be really anal, what they would do is kind of get that protein, two proteins that are only 5% similar in sequence, and see if they catalyze. This is actually pretty important for a project I'm working on in structural biology, because it seems that there is a code, and this is well known, no one wants to acknowledge its real implications. Oh, bless your.
I'm all right.
I know, bless you. I had the flu when I was a kid before they had the vaccines, and I've been vaxxed to the max for flu. Just because the symptoms are just so severe. The one time I forgot to get it, and I was so here, I don't remember ever being so sick. So I said, I do not want to go through this again. So just for those, I'm just saying that for those who say I'm an anti-vaxxer, I'm not. But I don't, let's go, Brandon. I don't believe in all these vaccine mandates.
I didn't say that. Sal's being the controversial one tonight. That's normally my department.
Well, just for you to, Woodridge, Virginia is the hometown of the Brandon.
That's true. That is true. That's true.
So anyway, I got on. Oh, because you were coughing. Yeah. So that 5% was just. Oh, I'm working on a project where we're actually able to predict 3D structure. So if you take all these sequences across species, how is it that you can predict 3D structure? Because if you remove, like, say, entire segments of the bacterial population, you don't get that. It's like every species has a piece of the puzzle. It's like every species group is needed to help define that particular created kind as far as a protein. So you have created kinds in terms of the species, but then you have created kinds in terms of the protein architecture that is spread across the species. And you can actually, this is like voodoo, that you could do an alignment, you run it through this thing called the direct coupling analysis, which is a nasty algorithm, and it's going to give you the 3D structure. And I was like, that's voodoo. And I'm going down to Dallas to talk to some people, a specialty in that. So the evolutionist said, well, why would God make it that way? And I'm just like, well, look what it's doing. This is like a secondary code that's just optimized to help you understand your own biology. I said, if you thought about it, and I've said this is why there's diversity. God didn't just create us and no other biological creatures. For us to understand ourselves, we have to sacrifice the lives of innocent animals, just like in the Old Testament, to find their healing and their atonement.
Yeah.
It kind of expresses itself in modern medicine, because the way we understand our biology is we take rats, cats, and primates. Sorry, Erica. We have to sacrifice them, and we understand our own biology. But then now in the era of genomics, we're able to do this. So where I was headed with that is, you were saying proteins might be the better avenue, and you'll actually be able to get homologies that you wouldn't be able to get in genes, because when I put in the NILB gene, I got like 20 hits. And I was it's like, this isn't going to do it. Dr. Sanford is excited. He said, oh, it's ancient. I'm like, we only got 20 hits. We need to get thousands. And then I did the protein blast and I got thousands. I was like, breakthrough. And we already put it on the preprint. Editor has already accepted it even before. He said, just reformat it. So we're going to get accepted again in peer review for that paper. Yeah. So I can walk you through the protein blast and some of their tools, and they're really cool too.
Okay.
If you'd like.
Definitely. I'll take that. I'm not gonna. I'll take free help wherever I can get it. So.
All right.
And I think it'll be helpful for our viewers too to see how this process works.
Okay. If I could just pause.
Sure.
This is really fun. This is really fun for me, that we could talk about this stuff, and I could also talk about a little bit about my work. But you're talking about baromenology with the DNA.
Mm-hmm.
I think one area might be in cellular architecture. We had to talk together on non-DNA inheritance.
Mm-hmm. Yes.
And there are cellular differences, like in the, I think it's like the centriole or something.
Mm-hmm.
That is not coded in the DNA. There are physical structures. The conformation is inherited. And that might be an interesting way to also define where there are boundaries. Like, for example, the way we're able to distinguish eukaryotes and prokaryotes is usually not just by DNA alone. It's just the cellular structure.
Cellular structure, right.
So, I mean, that's kind of very, that's a little bit too coarse. It's like you could tell a plant from an animal cell because the plant has certain organelles.
Right.
I don't know that, you know.
Could you tell an angiosperm cell from a gymnosperm cell without looking, without?
See, that's, I'm just throwing that out there.
Right. And that's a good question. It's something that could be.
Because the one thing that I could not, I mean, I couldn't, if I didn't know in advance what the human topoisomerase was versus, like, say, a plant topoisomerase, I couldn't just distinguish the two. So it's easier to be able to maybe distinguish it on kind of the more macro level. But so what's Gordon Wilson's approach? Would you have to be doing a set of genes or using taxonomically restricted?
His paper's not entirely clear. It seems to be he's looking at genomic equivalence and looking specifically for at the gene level, not at the sequence level, I don't think. Because if you went to the sequence level, it'd be the level he's proposing is just, you couldn't do it. And I think he probably knows that.
By the way, like, Gordon Wilson graduated from my undergrad.
Oh, did he? Oh.
George Mason. That's a creationist factory. Timothy Brophy, Timothy Standish, Gordon Wilson, David McQueen, Carolyn Crocker, Charles Jackson, and yours truly.
I'm sure George Mason is just thrilled about that, Sal. But no, what he's looking at is genomic equivalence, and to me, I think that there's potential in this approach. I'm not, some of you watch the video and go, he said that on the live stream. Yeah, I did. I think there's some drawbacks to it. And I walk through all that in the video that's going to go up sometime later in the week, probably on Sunday or something like that. But I think I'm wondering if maybe protein sequences might be more useful, or potentially even a combination of DNA sequences, because, like, with DNA sequences, you can't get 100% similarity just because of mutations and gene loss and all the other stuff that's happened because of the fall. So I'm trying to figure, like, how similar you'd have to say something was in order to say, okay, yeah, this is probably part of the same kind versus probably not. And you'd have to draw an arbitrary cutoff line somewhere, and I don't really like that idea.
And I need to point something out. And I got this from the dean of my school just by accident, because it was a cell biology class, but she just threw this out there on domesticated grapes. So let's say hypothetically, and you're the plant guy, I don't know if all grape kinds descended from one single grape, but even at this point in history, the domesticated grapes have already lost 15% of their homologous genes. On one strand, it's gone.
Right. And this is my, like, because I'm a plant guy, because I deal with polyploids, I know gene loss happens. It's a thing. You lose genes.
So it might be different. I mean, it could be, I'm just throwing that out there because we might have a created kind that are actually related, but it's like, oh,
right.
This lineage has lost so many genes, you're going to have a hard time kind of making, I mean, obviously physiologically they're so similar.
Right. And Wilson I think Wilson's maybe to account for this, he doesn't specifically say so, but he throws in a synapomorphies, like shared derived traits, as a additional, to go along with the genomic equivalence, which I think helps. I'm not sure it's the answer, but I think it helps. But like parasites, when we talk about parasitic organisms, they dump genes like crazy, but they don't need them because they're parasites. They are not free living. They just they're called parasites for a reason. They're getting their nutrients from something else, so they don't need a bunch of genes that they used to have. So they'll just dump them. There's no need for them. It just makes the genome smaller. They use less energy in replication. I mean, it's a win-win. They don't need them. So, and I think you'll see this with specialization, extremely specialized organisms too, is you'll see some gene loss.
Yeah. Wow.
Because you don't need them anymore because you're highly specialized for a particular thing.
So if I may say this is probably why I'm never. There are kind of two camps, not mutually exclusive. I put you in both camps. There's the baraminologists among the creationists, and then there are the anti-creationists. And Dr. Sanford and I are definitely in the anti-creation camp. We do not do baraminology at all. Don't touch it with a 10-foot pole, which is kind of fascinating. And then we have all, like I'll go to these creation conferences. We have these baraminologists. I'm just like, don't you like fighting? I mean, like I want to go up to Todd Wood. Don't you want to get in the face of evolutionists and just tell them what lousy science they have?
No, they don't, because they think they actually have good science. That's the problem. They think they do have good science. Don't get me started on that. I'll lose my voice. But no, you're right. But the thing is, somebody's got to take that baraminology back. They've had it for 30 years, and they've run it into the ground.
So, nothing personal, Dr. Wood, if you're listening to this.
You can take it personally from me. I don't care.
That would be you, Emory. So,
look, it's not a personal thing. I think it's not a personal thing. So anybody who's a Dr. Wood fan watching this, it's not a personal thing with Dr. Wood. I've never met Dr. Wood. So, I mean, I think some of his papers have rubbed me the wrong way. And I've kind of gotten the sense that he really doesn't have any interest in actually challenging the garbage that the evolutionists throw out there. He'll just accept it and run with it. And unfortunately, Sal is correctly characterizing the baraminologists as that is how they operate. One particular individual that I'm aware of, it basically says, yep, we can use and I did a video on this, we can use the word evolution because why not? No, because it has a meaning. It has a meaning, a cultural meaning that people understand. That.
And I wanted to talk to Matthew McLean and say, I don't know if you got to ask him, because he said, what, birds or dinosaurs?
Yeah.
And I said, okay, so if we extrapolate your methodology, does that mean we're Sarcopterygian fish too, human beings, and giraffes and birds too, or fish? I mean, do you want to go that far too? So I'd be curious what he has to say.
Yeah, it's a weird, and I know from sources that certain individuals in that camp are. Anyway, I'm not going to go down there. I'm not going down there. That's not what we're here for.
Really, all right, I got to tell you this. This conversation is the closest I've touched on baraminology. That might sound disturbing to a baraminologist. I just do not.
Okay, look, I get it. You look at those charts and all the math that they throw up to try and say that they've done something that's really intelligent. No, they haven't.
Okay.
They've regurgitated a phylogenetic tree using distance methods. Fine, okay.
So why did I choose my route? Because I'm lazy. It's just so easy to just put the flamethrower on evolutionary biology, because they just have so many flaws. I'm trying. You mentioned my series with Rebecca, how this conversation came about. We were talking last night because we were recording a show. I said, Rebecca, it's going to be limitless. We could just show all the problems, and we're not going to run out of things to criticize evolutionary theory. So we did ion channels. Then I started talking about the proteins involved in replication. And I said, this is problematic for the eukaryote prokaryote transition because they have to reformat with nuclear localization signals. And I said, Rebecca, we're not going to run out of things. And she's like, oh, yay.
She's got content forever.
So when I look at baraminology, I'm just like, I have to work so hard to just kind of just find one thing. And, you know, all I have to do for the anti-creationist side is just let's just comb through the chapter of my biochemistry or cellular biology textbook. Every 3 pages, I'm going to find something devastating. You know, I said,
But honestly,
I got it easy.
Not
so,
to be
sir,
mean to
let me just give you my salute. What you were doing, sir.
Not to be mean to the baraminology camp, but that's kind of what it's like reading through their stuff. Every 3 pages, you find something that blows up. I'm not even joking. Like the actual theory papers, like it's like, okay, that don't work, but okay.
And before I forget, just put this, file this away. And I know you have your notebook of things to you file away to visit later. We did talk about who was it? Kurt Wise worked for Richard Lewontin in Harvard. Lewontin he gave that paper that talked about morphospace.
Ah, yes, I'd forgotten about that.
And so Mr. Morphospace was not, the original Mr. Morphospace was not Kurt Wise. It was his mentor, Richard Lewontin, up there at Harvard.
I'd forgotten about that. I'll have to look into that.
So you're talking about all the garbage you read.
You send me more garbage to read. Thanks, Sal.
Okay, so viewers here, you're starting to see some of the dirty laundry in the creationist world. I uncovered the stuff on Kent Hovind,
like every 5th
and
video
Cindy Lincoln, and now we're even. I'm sorry.
I said that's like normal. If they're normal viewers of me, they know this stuff. I've thrown it out there before. It's not.
So all the dirty laundry's out, you know, from all the scandals, and then now even the theory. Oh boy. Anyway.
Yes. Anyway.
Thank you for all the information. This is a delight to me. But now let me be of service to you since you invited me to talk about browsing.
All right.
So we're going to switch now to protein browsing.
Yep, protein blast.
And it is definitely more stable, and I think it's not quite the headaches of gene browsing because you get chromosomes thrown at you, and the protein just so much simpler. So if you could bring this back up.
Yes, there we go. We're back up.
All right, so this is where we left off. That was the interferon, and this is where the whole thing of this is like a pseudogene, and it's interesting. Just to close this out before we move on to proteins, there are some humans where some individuals have a critical insertion in them that will reactivate an exon, and that gives them immunity to colds. So the fact that some of them are able to reactivate it, if we see that same pattern in the primates, where some of them have the pseudogene and then some of the primates also have the gene, that would suggest that these are just hotspots, that the cause of the common error is just that it's a common fragile point. And I think that would be important.
Yep.
Because then that's one thing less in favor of common ancestry. But it still did bother me. Okay, so I'm just going to leave it at that. I just never had time to go back and revisit this because there's so many things to move on to. So the most important database that I would go to, there are two of them, but the most reliable for our purposes would be UniProt or UniProt, as in Universal Protein Database, uniprot.org. And this is based in Europe, has some NIH funding. The other database is Protein Data Bank. I don't know off the top of my head what the URL is for that. Emory, can you?
I'll look it up. I'll look it up.
Can you put our faces back on here and just
Sure,
kind
one second.
that way I can see what I feel like I'll be interacting with a human here. There's a mode where you can shrink our little
There we go. Like
there.
that.
Yeah. So this is the most important. So like if I'll tell you one thing that's kind of like aggravating, unless you are kind of in the know, it's very hard to look up an individual protein because they'll require it under some sort of official identifier. So like if I typed in topo, like topoisomerase, I don't necessarily know that I'll find what I'm looking for. One would have to know that it's top 2A. So sometimes you do have to know a researcher and say, hey, what is the official 4-letter identifier for this? For example, when I typed in nyelinase, when I was doing this research on nyelinase, let's just type in nyelinase. Yeah, see, you get 3 hits, and I think we're lucky that it even gave this many because sometimes it won't. Because that will be the colloquial name, and you have to get lucky enough to find out what the official name is. So I'm just pointing this out. So what do? It's like, okay, you just go with what you have, and so you have a gene name here, and then you start to look it up. And so I'll look up this flavobacteria, and then I find out the gene name is somewhere here. Does it have a gene name?
Yeah, NYLC gene name.
Yeah, let me go back. I'm sorry, I should have gone to the entry here. I'll go to the entry. And there it is. The gene name is NYLC with a capital letter at the end. The protein will have it kind of the reverse with the capital letter at the beginning. And that's how you can distinguish when they're talking about the gene versus the actual protein sequence. So the gene, and it's going to be annoying when the gene has a very different identifier than the protein coding sequence, just for your information. And then you have all sorts of information here. But the important thing is you get the sequence, and you can click and you can get the FASTA sequence. And this is how I did the detective work for my paper for Dr. Sanford. I just went here.
Now, Sal, let me pause really quickly. Just real quick. The FASTA sequence is what you will need if you want to put it into just about any kind of program, right? So if you want to put it in Mega, you want to put it in Eugene, you want to put it in fill in the blank, you'll need a FASTA file. So that's very important that you remember, get the FASTA file.
Right. So now if you want to do searching, this is where BLAST comes in. You can type in, now that I have that, now that I have the protein, the amino acid sequence, I can go to BLAST, and then you use BLAST P. There's nucleotide and protein BLAST, so I go to protein BLAST. And then I just paste the sequence in. And there are parameters you can adjust, whether you want to search for 500 or 20,000. The search for 20,000 is going to be just gigantic. Now, the nice thing, what I was saying, the nice thing is you may have a bazillion strains of E. coli. It's just going to give you a few of them. It's the algorithm's smart enough not to torture you, because if it grabbed every sequence under the planet of E. coli, oh my goodness, that'd be kind of scary. So the curators of the BLAST database were like, okay, we're not going to do that. That's just going to, most people are interested in the consensus sequence of individual species. Don't give every strain, because the query results would be just totally unusable, because then you might get 50 million E. coli when you're really interested just across species. Right. So let's see what we get. We get, the default is 500. And you can see here it has the names of all the.
Yeah. Sal, I'm going to bring that up to the full screen just so people have a little bit easier seeing it, because that's small print.
So these are bacterial species, so you don't have to worry about exons and introns. And they do have some occasional repeats. because you'll see Agromyces mentioned. So they do have the strains, but it tends to be limited. And there might be a parameter where you could set so that you don't get too many repeats. You want to get a very broad. So you could see here, it did a good job of going to ones that had 60% similarity only, all the way to 100%. So if they had included like all the possible strains under the planet, you would not have been able to get this kind of nice overview of how that particular sequence is distributed. And as I said, the percent identity could go down to 5% and it's valid. We were talking, can you give us our little icons again?
Sure, 1 second. That's not what I wanted. That's what I wanted. There we go.
So where was I? I'm having a Joe Biden moment. I'm forgetting what my point was. I think first off, I don't know of any automated algorithm. Like if you wanted to do a survey across species, there's no way that you can just like download all the sequences that I know of. I've had to, if you find this out, that'd be very interesting. I've actually had to go here and go to each accession number and the entry, download the FASTA, go to the next species.
Unfortunately,
and you might be horrified to hear this, brother. I think I did that for all 500 once for my paper.
I'm not horrified because at one point for a baraminology study that I was running, I sat there and did 20 separate species, 300 characters each by hand.
Oh! All right, readers. Now you know what life as a graduate student is like.
Yes, welcome to being a graduate student, everyone.
I got to give an anecdote. I need a little comic relief because this is too stressful going through. Little comic relief. At least this is the kind of graduate work I could deal with. There's one guy, his graduate work, the reason I know about it, he was with the NCSE, the National Center for Cell Evolution. His PhD dissertation was on the length of penises in mice. And his whole dissertation research was taking his little tape measure. Okay. And okay, that's
why would you make that the focus of your PhD research? What meaningful point does that contribute to society?
Okay.
Anyway, go on.
So my PhD dissertation, Dr. Sanford said that what I did for nyelinase would qualify as a dissertation, and I appreciate hearing that from him. That's more of what I would appreciate doing because we overturned 2 papers, one in Science and one in Nature.
And for the record, for those of you who are not quite as tuned in, those are 2 of the top biology journals. You get published in either of those. It's a lifetime achievement for most people.
So thank you. We're going to publish in the lowest possible journal just to make a point that, falsehood will be in a top tier journal. The truth will be that overturns that falsehood will be in the most ignored journal. Mm-hmm. And I'm proud of that. Because
the top tier journals won't touch it.
Yeah. But one other graduate thing, and I was actually trying to see if Emory and I could talk about natural selection versus sexual selection.
That'd be a fun chat.
Because they used to say sexual selection caused the increase in brain size and capacity because women kind of liked the men that were more intelligent than the one that were burly. And it's turning out the actual studies are showing natural selection. Intelligent women don't have as many kids, and therefore the inference is intelligence is going down. So there you have it, one of Darwin's classical theory, 2 of them. Sexual selection is just kind of out the window with that. And natural selection is not building complexity. The actual data, as always, is crushing evolutionary theory. And we wanted to have a talk about that. But it just so happened when I was researching this whole thing about both sexual selection and evolutionary biology and courtship among males and females, I stumbled upon graduate level research. Some guy discovered that a certain mammalian species, like a mole or something, it copulates for like 12 hours, and then the male of the species dies by having too much sex. And I said, okay, so you're a graduate student. What are you going to do? Are you going to watch the two little moles, and you got your stopwatch, and you just got to sit there in your laboratory? That is not graduate level research that I want to do. Better someone else than me. I just had to throw that out.
If you think 12 hours is a lot, there's a species of aquatic isopod that I know about that does it for 3 days.
And I've heard one for 10 weeks. There's the book, Dr. Tatiana's Guide to Sex and All Creation, written by Olivia Judson. It's actually pretty interesting. So I just stumbled on that. You never know what sort of rabbit trails you're going to go down when you're when you may have just started off and just gene browsing. You don't know what kind of crazy things you run into.
This is why I bring you on, Sal, because we end up talking about stuff that's just completely off the wall from anything that was on the table. So I may have to put a warning on this for parents, though.
Yeah. Yeah. You know what's going to happen? I'm going to put that when I rebroadcast that everyone's going to watch.
Mm-hmm.
Gonna be
anyway, back to the genome browser.
Yeah, back to the genome browser. So how we got on this was the agony of graduate students, because I don't know that there's an automated way to do this.
There isn't.
And sometimes they will hire a graduate student that like really knows how to like kind of build his own web interface, and it's going to go there and grab it in an automated fashion. Now, when I was in my bioinformatics class at FAES NIH, they did talk about these APIs, application program interfaces, and you'd have to go to the NIH seminar. They'll teach you how to, you have to be a programmer. They'll teach you how to build these interfaces so you could go right to the database, and you could probably do these queries automatically. You'd have to be seriously funded to be able to do that, you know.
Yeah.
So usually that would be a research team that had a biologist and a dedicated computer programmer. I'm just needing to point out what the competition might be like. Just fair warning. But for cursory stuff, UniProt and BLAST will probably be the bread and butter. So let me just show a couple other things with this. So we did the BLAST sequence. I'm going to show you some cool things. What did I do? So I'm going to go to the accession number here. I hope I get the right guy. Endotype 6 amino. So this is the nyelinase. Now, you can identify conserved domains, and this is what's really cool. So I'm going to identify conserved domains, and you get this position-specific matrix thing. And this may sound kind of funny, but what'll happen, let me see if I can expand this. You have like a consensus sequence that they consider like a consensus sequence. That's kind of like the archetype that they decided. They'll pick out one particular species and say that's our archetype. The legitimacy of that's, you know, we can debate.
There are some significant evolutionary assumptions going into that, but anyway, go on.
But what they can then do is they're going to do an alignment there, and then they do a bit score. So that would be kind of like random probability that it could be arrived at. So a bit score of 313 means you raise 2 to the 313th power. So or the minus 313th power. That's the improbability that the similarity could happen by chance. So even with 5% similarity, you'll get these bit scores. And so it actually, the nice thing is it's going to evaluate it for you and tell you, is this good?
Mm-hmm. So what's a good bit score and what's a bad bit score, and what's the e-value mean?
Okay, according to the ID proponents, the good bit score would be around, say, 500. 500 bits would be like outside universal probability, but 300 is pretty good. Now, the evolutionary biologists, when they're really trying to squeeze some homology out of something and they're desperate to find similarity, they'll lower that bit score real low. And there is a relationship between the bit score and the E value. The bit score is kind of more straightforward.
Okay.
So we were able to do our paper with the 313 bit score. And so the important thing is when we have identified the name of a protein, it's based, when we're doing we talked about gene prediction. So a lot of times we're not actually able to extract the protein from the organism, run it through a mass spectrometry. It's very, very expensive. So a lot of the times when we say a protein exists in an organism, it's only because we've sequenced the DNA and we've done this similarity thing and said it must have that protein because look at this bit score. But strictly speaking, it's not the most rigorous establishment that such and such an organism actually has and expresses that protein.
The more I learn about genetics, the less rigorous it sounds.
Well, there are pluses and minuses to this because in a multicellular organism, you may not find it in a particular cell type. So I know definitely for human beings, and I talked to Paul Nelson about this. I said, have we actually confirmed orphan genes expressing proteins? He said, most of the cases, it's only the gene. We never confirmed the proteins. And he specifically cited the fact that it may not be expressed in the cell because the way a lot of cells, at least in humans, are differentiated is shutting down a gene.
So, and that's going to be true across most forms of life. You're going to have differential cell types depending on where you are in the body of the organism.
And there's one famous case where there was one gene that was expressed for only 3 hours in development, never through the rest of the lifetime of that mammalian species, which is just phenomenal. And that they found it is really incredible. So
I guarantee you there's more than one. I guarantee you that there is more. I think Sal may have died here, but I guarantee
more than one.
There's more. I guarantee you there's more than one of those genes out there that's expressed for only 3 hours.
Amazing. Okay.
Yeah, I was just saying, like, if there's one, there's probably several dozen that we just haven't figured out what they do yet.
Right. So I'm kind of giving a balanced view here, is that just because we haven't found the protein, it's like, well, there are an awful lot of cells in God's creatures. You may not. So it might be merciful that the DNA, that we have this homology that can tell you, hey, this thing might actually be there. You're just not detecting it in that particular cell type. So this position-specific matrix. So how I got there, I started off with BLAST. I went to the gene card, or what do you call this? The gene entry, and then you just kind of click around, and you mouse around to anything that they have here. And sometimes you run into, see, all I did is, and this is how I found the paper. I just clicked around, and I got to tell you something. I think if my professors were actually working on the BLAST program in the database, I don't think even they knew quite all the features. Because the NCBI, which is part of the NIH, is so gigantic. I don't think there's anyone that really knows all the ins and outs of how to get to things. And so usually you'll discover things because you know the researchers that worked on it, and he'll guide you there, but then he's not specialized in other things. So this will take you to the conserved domain database, and it'll take you even down more rabbit trails if you're really interested, like me, in proteins. But there is one thing that is nice about this gene entry. I think there's somewhere here you can actually go to the DNA sequence occasionally. Here. See, let's look up nucleotide. See if there it is, and see if we get the nucleotide sequence somewhere. And see if this is a nucleotide sequence. Oh, wait. Yes. So they'll give you the protein, and then I think somewhere you can for like bacteria, it'll take you to the nucleotide sequence. I'm not 100% sure. He says coordinates.
I think the genome sequence might be at the bottom, Sal, if I'm remembering correctly.
Okay, let's look. The gene. Now,
Yep,
there it is. Now back, prokaryotics, so nice. You don't have introns to muck things up. I don't know if it'll do that for plants. So kind of the nice thing is they did try some integration of the protein databases to link to the nucleotide databases. And that'll help. And then you could blast the nucleotide sequence if you really want to torture yourself.
I know what my project is for tomorrow.
Yeah. So I think the nice thing about BLAST is it does a lot of the curating for you, because heaven help anyone if they actually have to go through all the nucleotide sequences manually to try to do a comparison. Yeah, that would be, yeah. Praise the Lord for computers. And there is one, yes. There are curated databases and uncurated.
Important.
BLAST deals with uncurated because, sometimes, and this is understandable, a researcher might be the only guy that would even look at a particular organism. He's going to run it through his gene sequencer. He gets the data, he'll dump it for the cause of society. But it's like, it's an as-is basis. You don't know what you're getting. It could be contaminated. You have no clue. And so it's like caveat emptor, which you may have gotten. And then same with the gene prediction. They will provide the gene prediction for you because some of these sequencing machines, you could take this down now because this is about as far as I can go. Some of these, what's the name of some of the sequencers?
Goodness, I should remember this. There's a bunch of different ways to sequence. I don't know the names of the machines offhand, but like
there are
Illumina. I think Illumina is one of them. I think.
Yeah, Illumina. So Illumina will sell you, say, hey, we'll sell you the sequencer, and they'll say, hey, we'll even put a gene prediction package there for you. So it'll sequence, and of course the Illumina sequencer's 100 base pairs. And they're going to stitch together the genome based on evolutionary assumptions.
Right.
And you don't know. I'm just cautioning you, don't 100% trust. Okay. What does help is if they really do have, it's good to compare if you want, like, say, God's truth, so to speak, or at least the closest to it that we have on Earth, for whether that gene, the mRNA databases would probably be really, if there's one available for that really will tell you that was a good prediction. Otherwise, they're doing you're relying on the evolutionary assumptions to give it, which is actually,
it depends on how deep the assumption goes. Like if your assumption is that cats and lions are related, okay, that's a fair assumption. If your assumption is that cats and fish are related, then we've got a problem.
So I'm just saying, I don't have access to the mRNA database.
Okay.
But if you can find access to it and you have doubt about a particular, that's probably kind of like, okay, if you want to filter out the good, find out if you can get the mRNA database for the eukaryotic species. I would consider that very strong at that point to confirm a gene prediction. So you get that Illumina sequencer, they'll throw in the gene prediction, then they will upload it, that laboratory will upload it to GenBank or whatever bank that BLAST uses, and then it shows up. So I'm just cautioning what's out there. I hope that maybe that's hopefully some new information to you, or at least a different perspective.
It's been very helpful. I appreciate it. It's actually been it's been very helpful. We did get a question. So I want to throw this up on screen because I promised I would. It's a 2-parts question. So once you've read that, let me know and I'll bring up the context of the question.
This is really interesting because in church we were talking about the difference between men and women, and someone said, "Oh, we differ by this many nucleotide bases," and I'm like, "How'd you get that number?" I really wanted to find out because if you just take the Y chromosome by itself, it's like that's what 20 million bases.
It's not
That would be about 2%. We would be. So I'd say the Y chromosome itself is men will have. I think I'm legitimate in saying not so much genes but nucleotide sequences. We would have. Let me see the size of the size Y chromosome. So you will see, in answer to that question, you will see different people will have different figures for the differences between men and women. Mm-hmm. Someone will say 6,000 different genes or something like that. I don't know how they got that number, but I could do a quick calculation here. The Y chromosome 62 million in size, so 62 e to the 6 and 3.3 e to the 9th, and I'll divide this divided by that, and I'll get some decimal numbers. 98% similarity. So
pretty close.
Just based on the Y chromosome alone, you'll get different figures from other people. So, right. So we're very similar to women.
Thankfully, it's kind of important for the survival of the human race that men and women.
Now I know where this is headed. If an evolutionary biologist is listening, he'll say, "Oh well, we're 98% similar to chimps." So we're as you know, a human guy is as different from a human female as the humans are from chimps. They'll come up with stuff like that. It's not the same, guys.
No, if you think that those two numbers are identical, take a walk down to the vocal zoo and try to have a conversation with a chimpanzee, and then go home and have a conversation with your wife,
right?
And see how well which one of them works out for you. Now, depending on what state your marriage is in, the chimp conversation might work out better for you than but you get what I'm driving at.
Okay, now I really do want to emphasize this. It depends on how you do the comparison. It's like I said, you could take two random books and say, let's just compare every word in the book. And you'll say, boy, every word in this book has, you know, a homologue in the other book, similar. So like, and you'll say, you could easily conclude that they're like almost 100% identical. And I got to say this, when they say 98% similarity between chimps and humans, we're talking about selected gene segments doing it just like the way we compared those two books. Because when you look at the architecture, it's different. And we briefly...
Arrangement matters.
Arrangement matters. And I talked about the differences in the centromeric regions.
Yeah. And we can also talk about the fact that they ignored significant chunks of both genomes when they did the original comparison.
Yeah. So just be very careful with when you hear the statistics being thrown out, you know.
Yeah.
It depends on the comparison method. So thank you for the question.
Yep.
Who was
I got one more here. He gave us another one right after we got through that one up there. This one's probably a little bit more for me than you, Sal. Although if you want to comment, feel free.
Do you believe it's possible created varieties of kinds like lion and tiger? Specifically for the lion and tiger, they can interbreed. So and Emory's more the baraminologist than I am. But anytime species can interbreed, it's like that's the same kind.
Yeah, no, and so my answer to this would be at the beginning there probably was variability built into the original kinds. Dr. Jensen talks about the created heterozygosity model, and I think that's probably true. But I don't think God created original pairs except for humans. That wouldn't make any sense. That would make absolutely no sense for. I mean, how would you could not fill the Garden of Eden with a pair of grass plants? Okay, so he's creating more than two. At least for some things, there may be some where he created original pairs, but I think for a lot of things, other than humans, it was more than an original pair. And if he made more than an original pair, he probably also made of those original pairs some variety, at least in their outward appearance. Now, there may not have been species built into the original creation. I suppose that's possible, but there probably was variety within the various extant kinds at creation. There probably was some variety pre-built in. It's just the only way it makes any sense. But yeah, that's a good question, and that actually shows that you're thinking about the concepts.
So any things that we're not sure of whether the same kind, if they can hybridize, we would assume it's like it did have a common ancestor as created ancestor. Now there are situations where two creatures are not able to breed, but they might still be somewhere along the way have a common ancestor. So I'm presuming what baraminology is trying to do is we're trying to identify what groups of creatures descended from one particular common ancestor, originally created.
Right. Although I will say that the current extant definitions of a baramin doesn't even talk about ancestry at all. The way they define a baramin is a potentiality region in 3D morphospace. Back to Lewontin again. Yes, I know. There's not a thing about reproduction in the definition. Not a thing.
And yet that's such, I don't know.
I hear you. I'm and it's completely divorced from anything Scripture talks about, which bugs me to no end.
But you gave me a new data point where they're just, since you're a plant guy, it's like I never thought about that. Would there be just one created grass blade to start with?
And I never thought about that. The other thing to think about when it comes to this, and I think about this kind of stuff a lot because I'm a nerd. The flood wiped out all the air-breathing, land-dwelling animals that didn't go on the ark, but it didn't do that with the plants or the insects, which leads you to potential answers for the question of why there are so many orchids. Because if even supposing Noah's wife said, you know what, I like those, I'm going to stick them in a pot and take them up to my room, and took some orchids with her on the ark, which that's possible. Women decorate when they go places. They like to decorate stuff. That just most women don't like that. And I guarantee you, Mrs. Noah had some plants on that ark somewhere, some flowering plants somewhere in that ark, because that's how things work. But even supposing she took a couple of orchids and then planted the seeds after she got off, because I want those outside my house when we get off the ark. There's going to be a bunch of orchid seeds, a bunch of even maybe orchid seedlings that survive, which means you're going to have a lot more variety to start with after the flood than you do in kinds like cats and dogs. And as you can see with cats and dogs, there's a lot of variety that was available to start with. And then add on to the fact that, we've got now created heterozygosity in plants, and then with the potential also for polyploidy, which increases the number of potential alleles that are available at any one point, is no. we're looking at a huge, which is why I think that when we finally actually do baraminology on plants, which most people don't, Wood's done a few, but I think of the ones he's done, I think he did like 20 or 30 or something like that. And he did a book of it where he did like 56, I think, different kinds. Of those 56, I think like 50 of them have to be thrown out because of and this is not his fault. This is, I'm not blaming him. It's just I sort of am blaming him, but I'm not blaming him for doing anything wrong. He didn't do anything wrong, but nobody's ever done any simulation studies to know how this works. And I have now done them. And I've gotten some help, and I've helped some other people do theirs. I've seen some other people look at stuff. And basically, if you don't have a taxa ratio of better than 2 to 1, a character to taxa ratio of better than 2 to 1, all you get is noise.
Wow.
And it doesn't matter. I've tried both programs, BDIST and Barclay. I've verified this because a friend of mine is into this, and he does a lot more of this than I do. And I've verified what he was talking about, and yeah, you just get noise. It's pure noise. You can get patterns, but there's noise mixed in with a pattern, so you don't know what pattern is real and what's false.
That's why I just salute you guys. I think that's one of the tough areas of creationist biology is baraminology. just, before we close the program, I did want to say there's one interest in, as you know, Emory, we are just so spread thin because there's so few of us. And like you said, even the evolutionists don't have the capacity to handle all the data that's coming out. And I've heard that same thing at the NIH, is that it used to be we didn't have enough data. Now we don't have enough ways to integrate and synthesize and analyze it.
Yeah.
Because it's just so much. It's just like I said, heaven help us if we didn't have computers and you're going to have to look through all the plant genomes to find homology. So
Anyway.
yeah. Does make us wonder at the creator who could make all these.
Amen.
The one area where, if I really did have time, I would like to be right there beside Nathaniel Jeanson to he and I have had conversations, and I was like, Nathaniel, I mean, it's not like I don't know population biology, but I didn't understand a thing you said. But I have a feeling, because I have a feeling that he and I could work through this, because there is, as you know, kind of this kind of this mortal combat between Dan Stern-Cardinal and Nathaniel Jeanson. And I've publicly said, I don't know. And it's not like I'm dodging. It's sincerely, it's like I'm in population genetics about as anyone is in the creationist community. And if I don't understand Jeanson's work and I don't feel 100% comfortable, I'm not saying he's wrong. It's hard for me I can't even defend what I don't understand, if you know what I mean.
Right.
That's
If you don't understand something, you can't defend it adequately. That's the way to do it.
And I'm like, I would like to frame it in a way that I can understand. And I just haven't had the time. But the gene browsing, I think, I have a feeling he's onto something. And you probably may have seen this yourself. Because when we talk about created heterozygosity, that would mean that specialization happens through the increase in homozygosity, I would presume. And I wanted, I'm glad I have you on camera now, or through StreamYard really, because I've been dying to ask you that for like the last 6 weeks. Do you think that's the way Nathaniel defines specialization is, it's a little different than the way others might, but it's basically through the process of more homozygosity. And I said, there's something there, because that's actually a prediction of where it's going, and there may be insufficient homozygosity at this point. At the rate it may be propagating.
Well, there's a prediction there, or a potential for a prediction at least, that if you get too far into homozygosity, you can specialize yourself right into extinction.
Right. And then there's another thing, and I noticed this across, and this is, people ask, why are you a young earth creation? Look at all the evidence of evolution. I just laugh at that. They're looking at all these, I was looking at these living fossils, and I'm like, oh my goodness, there's not hardly any sequence divergence in that entire clan in homologous proteins. I'm like, so let's say we're, I'm dying to ask Matthew McLean, are we all Sarcopterygian fish if you're going to say birds are dinosaurs? Let's just go all the way then, So let's say we're all hypothetically Sarcopterygian fish. Why is the coelacanth and the lungfish, why are their proteins so similar? And yet you have kind of 3 lines, one that would have the coelacanth, another with the lungfish, and then 20,000 different varieties through the tetrapod.
Right.
I said, this doesn't look right at all. And there's not enough divergence. And then I looked at this They're cycad plants
Yeah, I know what those are.
because they said these are ancient, these are living fossils.
Yes.
They did a sequence divergence. They said, oh, we're going to find the molecular clock goes all, you know, they took a variety and it's going to go back 300 million years. And it didn't. It was only like 1 million. And then Lenski did some clocking of mutation rates. And I looked at some papers. I said, well, we could be off by a factor of 200. So I said, okay, 1 million divided by 200. Let me do that. 1 to the 6.
Sounds like about, if doing it in my head, it sounds like 5,000 years.
5,000 years. You know where I'm going with that.
I know.
And I'm like, Nathaniel, we could have so much fun. You and I and Emory, we could have so much fun looking at this. So let me just throw this out since, okay, I'm going to try to, I know you want to fight that anti-evolutionary battle in baraminology space. I'm just putting a little bug in your ear that while you're looking at all this, look at the sequence divergence in homologous proteins. You might, if you can establish that you have an absolute kind, then you look at the polymorphisms in one kind. You might see this pattern because the evolutionists found it in Cycads, this fern-like tree. So, and I was interested in animals. And I'm just like, okay, this is going to be very interesting because I was trying to go through all the most recent common ancestor. I was putting it through some of these. It's so steeped in evolutionary nonsense that sometimes I can't tell what's good from the bad in terms of real phylogenetic predictions. And so, you know, I have Dan Stern Cardinal on one side, and I said, I think some of your arguments are good. And then I listen to Nathaniel, and then I read the peer-reviewed literature, and I'm totally confused. But it's like, I think we could, if I just had enough time, I'd go back into that. But there's just so many other research fields. You know what? I think I'm making a slight announcement. I might be bailing out of biology a little bit because my love is physics. And there's the distant starlight problem, and there's geology and the radiometric. That's where.
Now,
Right when I started
if you bail out of biology, who am I going to ask to help me with my homework? So, for the record, for those of you who don't know, because most of you probably don't, I took a evolutionary genetics class last fall. Got a pretty good grade in it at the end of it. But at the beginning of the semester, because there was so much math, I was really struggling with it because math and I are not friends at all. And so I rang up Sal and I said, hey, Sal, you would help me with my homework. Best grade I got on a homework assignment that involved math.
Well, I was, you know, I will say this. Thankfully, it was stuff that was in the papers that I written, and it was really obscure. But I remember, you're gonna laugh. I think when I was driving off to church, I kept thinking about that equation. It was heterozygous advantage. It didn't go by that name, but that was so cool. And I said, oh, I was wondering what that was describing. He said, that's sickle cell anemia.
You know, I didn't even think of that when I was working through those problems, and I know about sickle cell. I didn't even think about that.
Yeah, but they didn't use the word heterozygous advantage. But I was like, why are you putting that little, you know, that little coefficient there on a heterozygous form? I said, you're going to get, you get this kind of funny equilibrium of, you know, because normally you're going to expect one allele to dominate. Why is it you get kind of this equilibrium of a mix of alleles? And I realized, oh, that's heterozygous advantage. Sickle cell anemia.
Yeah, I didn't even think of that because I was using a different term, but I didn't even think of sickle cell. But yeah, I know. Yeah, I know what that is. Yeah.
So anyway, I'm glad we had this conversation because there are just, you know, if you identify a kind, you might look at the polymorphism profiles. They might tell you that the species is young. And especially if you happen upon a living fossil plant, like the cycads, I hope you publish on it.
Well, I've got some papers in the works that are hopefully going to publish at some point. So,
and I did want to tell you there are because of the internet, we're finding a lot of creationists we wouldn't have expected. Because I'm finding there's so many of them, but because of the internet, we have all kind of come out of the closet amongst each other.
Right.
And so there would be people like Ola Hofstra or whatever. So you could take the data and say, you know, can you tell us how far back the common ancestor is? And Ola has kind of old Earth models. So it'd be very powerful if someone like Ola would say, hey, you found a recent common ancestor. That would be really cool.
Yeah, I'll have to figure out how to do all the sequence divergence stuff. I'm not great with the genomic data once I've got it. I'm still trying to figure all this out. But between Eugene and Mega, and by the way, if an Evo watches this and knows of a program that's free, because I'm a grad student, I don't have money.
Yeah.
But if you know of a program that's free that like I should get that I don't have, because right now I'm working with Eugene and Mega. But if you know of something else that I ought to have that I don't have listed there, let me know.
So you certainly taken on a very difficult genetic area, stuff with polyploids. I'm just like, oh wow, I have hard enough time with diploids, you know.
So I'll just throw this out there. I am considering going for a PhD, and if I do, I'm thinking polyploidy might be where I go.
I had a hard enough time doing the genetics of a diploid, even a haploid. I mean, so where was I headed with that? If you have any interest in animals, again, you might be able to do the clocking, and so you know, they keep, you know, the other side keeps trying to write off the young Earth creationists. It just does not go away because it looks like it's written in the genes and in so many other areas across, you know, the marks of this are across the universe. You know, people wonder why. It's like, well, you know, you've heard part of it right here.
Yep. So, all right. So we've been for 2 hours. Thank you, Sal, very much for the help. I learned a lot. Hopefully our audience learned a lot. Audience, you've been great. Thank you for the great questions. I appreciate it. Thank you for putting up with Sal and I nerd out for 2 hours. And I hope to have everyone has a great rest of their week. And I look forward to seeing you all the next time we go live. So thanks for joining us.
Thank you.